The overall goal of this image analysis software is the parallel analysis of protrusion dynamics and relative protein concentration along the entire filopodial length.
This method can really help us understanding key questions in cell biology, especially the individual proteins involved in extension and retraction of filopodia.
The main advantage of the technique is that the adaptive shape tracking algorithm provided allows quantitative assessment of protrusion dynamics and spatially resolved protein localization.
To begin, culture neurons, HeLa or CUS cells in supplemented medium as detailed in the text protocol. Once 40% confluency is reached, transfect the cells with the constructs of choice using a transfection reagent as per manufacturer's instructions. Keep the transfected cells in an incubator at 37 degrees Celsius and 5% CO2 for 15 to 18 hours. Following incubation, fill the cell culture chambers up to 90% with 37 degrees Celsius pre-warmed medium containing 20 millimolar HePs to reduce changes in pH and osmolarity due to evaporation during image acquisition. To seal the lid, apply a thin layer of vacuum grease on the inside of the lid and gently press it on the culture chamber containing the transfected cells.
Possibly the most important part for image analysis is the image quality. One thing we have to take care is the signal to noise ratio, which has to be more than 4. And we can ensure that by adjusting the exposure time of the camera and the laser intensity.